Hi, everybody. Today's lecture is going to be focused on senses. We will be discussing your sense of vision and auditory in a little bit more detail, and your sense of taste and olfactory briefly. Sense of taste is basically, or your gustatory sense, is associated with about 4,000 receptors that are found on the surface of your tongue. And they give you your ability to sense the taste of bitter, salty, sweet, sour, and what we identify as umami. So these are basically, again, these senses associated with little taste buds, which are the elevations on the surface of your tongue. Now, you already kind of, I hope everybody's familiar with the typical senses, but umami might be relatively new to many of you. So umami is what we identify as sense of savory. So when you have food that are rich in something called monosodium glutamate, which is commonly known as MSG, you get to feel that food tastes really good or delicious, or as I said, savory. Now, normally you will find MSG in the correct amount, or normal amount, in cheese, beef, and seafood, as they are rich in certain group of amino acids. However, a lot of times industries introduce MSG at much higher quantities into their food to make something that may not be as quote unquote savory to taste better than what it is. And a lot of times we do have that craving for outside food because, again, normally you don't add MSG to your food versus if you buy food from outside, they're usually pretty high at that component. Now, in terms of the location of the taste buds, we do have specialized areas on your tongue that gives you the ability to basically sense or taste a specific taste, like bitter, sour, salty, etc. However, do understand that even though this picture is there, that tells you, for instance, your taste of bitter is at the back of the tongue or basically back of your throat, that doesn't mean you cannot taste bitter with the tip of your tongue. All this means is that areas of your tongue has higher concentration of these receptors versus more or less distributed receptors in other regions. But overall, if you're looking at your sense of taste, 4,000 receptors compared to millions of neurons and receptors we have throughout our body, it's not necessarily the strongest sense that we have. On the other hand, if you look at your sense of a smell, you can have average of 10 to 20 million receptors associated with your sense of a smell. Therefore, your sense of a smell is much stronger than your sense of taste. And what we typically identify or perceive as taste, as in your food tastes good, is associated 80 to 9% with your sense of a smell. Hence, if you are sick or you have your sinuses blocked, the food tastes pretty dull because your sense of a smell is blocked. Now, how do I get my sense of a smell? It is associated with modified neurons that are found inside your nasal cavity, more on the superior aspect of it. And these are olfactory cells, and they have the ability to interact with molecules of odor and convert that to your sense of a smell. Here's kind of a depiction of these modified neurons. So the molecules of a smell can travel to the superior aspect of the nasal cavity, where you have extensions of your olfactory nerve. And that signal can be transferred all the way to your temporal lobe, where you will be basically perceiving your sense of smell. We also talked about this in central nervous system, that a lot of times a specific smell either trigger a good or bad memory for you. And the reason for that is because your olfactory bulb is directly connected to your limbic system that can activate and target the specific memories and activate those based on the specific smell you're sensing. That's about it as far as your sense of taste and smell. We are gonna move on to your sense of vision or your visual sense, which is going to be focused on identifying the three layers of the eye, looking at the anatomy of each of these layers, and then briefly look at how we actually create our sense of vision and pass that message to the occipital lobe for what we identify as our sense of vision. So your eyes is made up of three separate layers. These layers are identified as tunics. We have the outermost layer, which is fibrous layer, depicted in blue. We have the vascular layer, which is basically rich in blood vessels, allowing for gas exchange and nutrition exchange, or providing nutrients. And sensory layer, which is depicted in yellow, it is the innermost lining of the eye, also better known as retina, and that's the layer responsible for creation of your vision. So let's go ahead and take a look at each of these layers one at a time and take a look at the anatomy and then the function of each. So when you look at the fibrous layer of the eye, it is made up of two main structures. So what we identify as the white of the eye, which is this part, that's the sclera, that's the white of the eye. And then notice as it comes in front of the eye, you see the bulging of this layer. This is what we identify as your cornea. Now, even though cornea still have the functional property like a sclera, it is identified, it is translucent. So you can kind of see again the white of the eye transition into the cornea. So it is translucent, which means that light can pass through it. So light basically can go through the eye and then get into the internal structure of the eye. The main function of this layer as a whole is protection of the internal structures. Cornea, which is again the translucent layer, has two characteristics, which is not listed here, and I do want you guys to know. It's first of all avascular. Basically means that has no blood vessels, which is kind of a good thing, because imagine if you have blood vessels running right in front of the cornea, then you always will have lines running in front of your vision, affecting your vision, causing the blurriness of your vision. However, even though it has no blood vessel, it is highly innervated. And most of these innervation or presence of nerve is pain nerves or pain receptors, which kind of makes sense again. Kind of imagine you poke yourself in the eye, it hurts a lot. And the main reason is because you want to protect the internal structures of the eye. Now, the question is, if the eye or your cornea is avascular, where is it receiving its nutrients and oxygen? So cornea receive its oxygen directly from the air, which I think is fascinating. So here is the air environment, the cells of the cornea directly receive it. In terms of nutrients, it receives it from the fluid that runs in this space right here, and it is referred to as the aqueous humor. So it is the fluid behind the cornea, and again it is rich in terms of nutrients, and it can allow the cornea to collect anything that it needs, but no blood vessels. Now the second layer of the eye is the choroid layer. So I like to kind of depict this for the students, even though there is a picture, a lot of times it's easier for you guys to follow on what I'm drawing. So if this is the second layer of the eye, so this part of the second layer of the eye is called the choroid layer. So choroid layer has lots of what we identified as melanin. which is a color pigment. So why do I care about this color pigment? It allows for the extra light that enters the eye to be absorbed. So basically, it doesn't allow that light to kind of bounce around inside the eye, and helps you basically create a clean image. Again, if you're collecting any additional light, that light would not be bouncing off inside the eye, and it would make sure that the eye is doing what it's supposed to do. Now, as you come forward toward as you come more toward the front of the eye, you start to form these structures you see right here. These are a specific type of muscle called smooth muscles, or they're smooth muscle called ciliary body or ciliary muscle. And these muscles are connected to little strings that are coming off of this on either side. And again, there's a nice picture. I just like to draw it for you guys. And these strings are called suspensory ligament. So what are they actually suspending? What's the purpose, as the name implies? Right smack in middle of the suspensory ligament, we will have our lens. So here is our lens. So you have the lens that is connected to the suspensory ligaments, which connected to the ciliary body or muscles. So kind of imagine if I am taking the lens and I'm pulling the suspensory ligaments, the lens can be stretched. And if I relax these ligaments as I'm making these ligaments loose, then the lens is not being pulled on either side, making my lens round. So the change in the shape of the lens is extremely important, and I will talk about it momentarily as we go through the information. But right now, let's talk about and focus on the anatomy. Now, the continuation of the choroid layer as we come farther forward is the color portion of the eye, which is known as the iris of the eye. So the black portion, this is your iris. And right at the center of the iris, you have this opening, which is called the pupil. A lot of people think pupil is an actual structure, but the reality is that pupil is just the hole at the center of the color portion of the eye, which is the iris. So I can change the iris, and through changing in terms of the iris, I can change the size of the pupil. Iris is technically muscle. So let's take a look at this. Choroid, same thing as we discussed. Choroid is the thin middle layer of the eye. It is extensive in terms of blood vessels, so it is important in nutrients and gas exchange. It does contain the dark pigment, which we identified as melanin, right? And the iris portion of the eye, that's the continuation of your choroid layer, which is basically the color portion of the eye. It is made up of a smooth muscle, so please again do note that. And pupil is the central opening where the light passes through. You can change the size of the pupil by adjusting or either contracting or relaxing of the iris muscle. So here is our picture, guys. Choroid layer in the back, come in front. Here is my ciliary body. These are my suspensory ligaments on either side, right? At the center, you have your lens. Coming farther in front, you have your iris. And at the center of the iris, the opening, that's your pupil. So lens, what is your lens? Lens is a structure right behind the iris. What's the purpose of the lens? It literally angles the light. So it allows you to focus your light. So if you ever seen, sorry for using this example, but when you have the kid having Wow. A magnifying glass and it hands on it, focusing the lights to kill the ants. Think about you have very similar structure inside your eye, but with the goal of focusing the light and giving you the capacity to create an image. We talked about ciliary muscle, suspensory ligaments. Again, ciliary muscle connected to the suspensory ligaments, which holds the lens in place. Here again, depiction, you have ciliary muscle, suspensory ligament, and the lens right at the center. Now, why do I even want to change the shape of my lens? The main purpose of changing the shape of your lens is to be able to focus on objects that are near me versus objects that are far away from me. So if the objects are close to me, the way I want to do is I want to make my lens thick or more rounded. So with the help of the ciliary muscle, I'm going to let my ligaments to relax. So the ligaments that are holding my lens on either side, if you kind of imagine it again, these ligaments, here's my lens, and here's my ligaments on the other side. If they are loose on either side, my lens is not being stretched, so the lens becomes more rounded and allowing me to focus on objects near me. But if I am pulling on these suspensory ligaments, my lens can become really thin, and it becomes thin, it allows me to focus on far distance objects. So here's a depiction again. Look, strings are really tight, right? Lens is stretched. Look at the thickness of the lens, not that thick. Ligaments are loose. Lens is rounded, allowing you to focus on near, again, versus distant object. This is known as visual accommodation. Now, when you look at your eye, the third layer of your eye is known as retina. Retina is extremely important because it has two types of cells called photoreceptors. Break down the word for yourself, guys. Photo means photons of light, basically. Receptors as in sensitive to, right? So photoreceptors means basically you're sensitive to light. Now, what are these two types? Why do I need two types? Rods are the cells that are going to help me see in the dim light. Now, when you're in a dim light, guys, do you see a lot of details? Probably not, right? So this is just gonna basically give me where the, let's say, the furnitures are in the room. You give me kind of a shade of a gray where things, again, located, but I don't see a lot of color. I don't see a lot of detail. There's no vividness or acuity to my picture. Cones are going to give me ability to see in the bright light, right? So this is the one that's gonna give me what? Clear, colorful images. Sorry. This is the part that is basically going to give me the capacity to create beautiful images when I walk into a lighted room or when I walk outside. So once again, rods are going to be very sensitive to light. And this is the part that confuses students typically. I said light is gonna give me ability to see what? In the dim light, correct? So how is this that means they're sensitive? Think about it. When you're in a dim light, do you have a lot of light or little light? That means little light. But what is that little light doing to those rods? It causes them to activate, right? Which means it doesn't take much light to activate them, making them what? Very sensitive to light. You only need very little light to have them activated. What's their shortcoming? They really don't give you much detail. There is no color, there is no vividness, again, no acuity into your image. It gives you the ability to kind of see the shades of the gray in a darkened room or at night. Now, cones, on the other hand, they are not very sensitive to light because you need to do what? Give them, kind of again, think about them, give them lots of light to what? To activate. So you need to give them a lot of light, which means little is not enough for them, which means they are not sensitive to light. But when they do get activated, they're gonna give what? Give you the ability to detect color. They allow you to distinguish between colors, and that basically gives you the capacity to see color, vivid images in an environment. Now, you do have 3 different type of cones, and these are red, green, and blue, as you can see as your larger circle. And when these circles overlap, you get the remainder of your color. Now, in the back of your eye, you do have an area, typically located right about here. And this area is referred to as the fovea centralis. And this area is where you have a densely packed cones, no rods. So this is the image that you want to create. When if you want to create an image, this is a point you want to create your image at, because it's going to give you the sharpest, most clear image possible. So when you look at your vision, up to this point, we have said that your rods and cones are the ones that are identified as your photoreceptors, as in they are the ones that are going to give you the ability to create image. Now, within your retina or your sensory tunic, you don't only have rods and cones, you actually have 2 other layers of the cells. So what is the job of your rods and cones? And I'm going to kind of depict this for you guys in a slide. So here is my thing. I know it's already in your thing, but I'm just gonna depict it for you guys. So at the most basic, all the way at the back of your eye or back of your retina, you have what we identify as your photoreceptors. And these would be your cones and rods. What is their goal? Take the light, okay, and you're gonna convert it to electrical signal, okay? So that's the job where the conversion happens. Now, the next, oops, sorry, I think I lost my picture. The next part of this process, so I'm just gonna show it on this. So here are my rods and cones. So the signals created, now they're gonna pass it to the next type of cell, which are identified as bipolar cells. So rods and cones create the signal, they pass it to your bipolar cells. And then bipolar cells take that signal and pass it to the 3rd layer, which is called your ganglion cells, which are these red cells you see. Now, these are neurons, right? So look, you have your dendrites, you have your cell body, and then coming off of the cell body, you have your axons. So these are all axons, right? Now, when these axons are merged together, these long axons, when they merge together, they form what we identify as optic nerve. So optic nerve, if I say, what is optic nerve? Optic nerve is what? Axons of all of the ganglions that are joining together to come basically from the back of the eye. Now, what happens after that is you want to reach from your optic nerve, you want to go all the way to the occipital lobe. But remember the whole understanding of contralateral. If these words sound unfamiliar to you guys, please go back and review your central nervous system. Contralateral means left controls right, right controls left. So at this point, the signals needs to crisscross, right? So the signal from the right eye needs to go to the left and vice versa. So here's my eye. Here are my optic nerve. This point, when they're crossing over, that would be your optic chiasma or optic chiasm. And then they continue to go farther back and eventually reaching my occipital lobe, which is where your signal will be process and give you your sense of vision. So looking at this, the visual pathway starts with rods and cones. Signal gets passed on to the bipolar cells, which pass it to the ganglion cells. Axon of those ganglion become the optic nerve. Optic nerve continues and crisscrosses at optic chiasm or optic chiasma, and eventually ends at the occipital lobe, which is the posterior aspect of your brain. So I'm going to stop here for you guys, and we will continue with the senses lecture for our next one. We're going to touch upon bases for the eye, and then also discuss the ear and our sense of hearing in a little bit more detail as well.